So any substance that can be chemical or physical parameters which causes mutation to an organism, that are called mutagens. Okay, so mutagens are of two types, that is physical mutagens and chemical mutagens. Physical mutagens and chemical mutagens. Physical and chemical mutations. Okay, and physical mutations are of again two types, that is it can be either temperature or radiation. Temperature or radiation. Any substance which causes mutation to an organism, that are called mutagens, and mutagens are of two types, that is physical mutagens and chemical mutagens. Physical mutation means it may contain temperature and radiation. Okay, first we will discuss about what are physical mutations. When temperature increases, the rate of mutation also increases. The rate of mutation also increases. When temperature increases, the rate of mutation also increases. Okay, so that's how temperature. Then what are radiation? Radiation are of two types. Radiation are of two types. Okay, that is ionizing radiation, non-ionizing radiation. Ionizing and non-ionizing radiation. Ionizing radiation include alpha, beta, gamma and X-rays. Whereas in non-ionization it includes UV rays. In non-ionization, UV rays is there. So in higher organism, our skin have the ability to penetrate the UV rays, so that higher organism won't absorb these radiation. Okay, so in higher animals they don't cause any effect, but UV rays and radiation are effective mutagens in microbes. UV radiation and UVs and other related to the radiations are more effective, and it leads to the death of that particular microbes. So in case of lower group of organism or the microbial organism, these UV rays are harmful and it causes death. But in higher organism, mostly our skin have the ability to penetrate, and absorb these radiation. Clear? Okay. Then that's why we use UV rays in the plant tissue culture technique. Okay, plant tissue culture technique, that is in vitro culture, we will use these UV rays to kill the microbes, to make the area microbe free. And UV rays and nitrous oxide, that is HNO2, they have the combined effect of them are deamination of nitrogenous base pair. Deamination of nitrogenous base pair. Deamination means removal of amino group from the nitrogenous base pair. Okay, so due to the deamination of nitrogenous base pair in adenine, okay, when the amino group of adenine is removed, then we will call them as hypoxanthine. When the amino group of guanine is removed, we will call them as xanthine. When the cytosine, that is the amino group which is present in cytosine, is removed, we will call them as uracil. Okay, so these things will happen when the amino group which is present in these nitrogenous base pairs are removed. We will call them as these particular names. Okay, then UV rays don't cause any deamination in thymine. Thymine, they can't cause any deamination. So these are about physical mutations. Then finally, chemical mutations. So compared to physical mutation, chemical mutation have the ability to cause more curse or more error in the body. Why? Because the physical mutations, they are mainly localized effect. Okay, the effect of physical mutations are localized in a particular location only it will affect. But in chemical mutations, they spread the complete body through blood circulation, and which they reaches the gonads and causes germinal mutation. So as a result of chemical mutations, germinal mutation may be happen. And chromosomal mutation also produced due to the chemical mutations. Chromosomal mutation. Germinal mutation as well as chromosomal mutation. Clear? Okay. Then examples are mustard gas. Mustard gas. Mustard gas is the first observed chemical mutation. First observed chemical mutation. First observed chemical mutation is mustard gas. Then carbon tetrasulfide, nitrous oxide. Okay, nitrous oxide, carbon tetrasulfide. Then DDT, dichloro, diphenyl, trichloroethane. Then LSD, that is lysergic acid diethylamide. Then ethyl urethane, pesticide, organic peroxide. So many type of chemical mutations you can see. Okay. Then when the antibiotics like neomycin, kanamycin and streptomycin, they combine with small subunit of prokaryotic ribosome. Okay. When the neomycin, that is the antibiotic, neomycin, kanamycin, and streptomycin. When these antibiotics, they combine with the small subunit of ribosome. Small subunit of ribosome, they will cause mutations in that particular organism by changing the reading frame in translation. Okay. Then same effect can be done in, so this may happen in prokaryotes. Same thing can be happen in eukaryote also. A antibody combined with a smaller subunit of ribosome causes effect. So the antibody is puromycin. Okay, so puromycin have the ability to combine with the smaller subunit, and it may cause the mutational effects in eukaryotes. Okay, so these are about mutagens.